Okay, okay. I'm sure everything is fine tonight. Is that so?
Yes, Doc. Clear.
Okay. So our topic is pesticide metabolism. Now, if we focus on the pesticide, it can either enter the plants or the animal, where it could be insect. Now, what happens thereafter inside the organism is that the pesticide is going to meet some enzymes within the organism, and these enzymes facilitate certain biotransformations, which we may call metabolism or co-metabolism. These are basically biochemical reactions. Now, if we target a pesticide to plants, the byproducts which are produced after the breaking down of the pesticide are incorporated within the plant. But when the plant dies, it may be acted upon by microbes. And then these microbes can work on the byproducts of the pesticides, and usually we may end up with the nitrogen and the carbon dioxide, or simply carbon and nitrogen will be formed in the end, probably oxygen. In the animal, the products which are immediately formed may be lost through excretion. And these byproducts, when they reach the environment, they may meet other microbes and they are further degraded. Basically, when a pesticide enters a living organism, it undergoes 3 phases of breaking down with respect to the plant. But in the animals or insects, we may have 2 phases, phase 1 and phase 2 of the pesticide metabolism. This is not actually new to us, because when we talked about herbicide selectivity, we touched on some of these issues, but we were specifically focusing on herbicide selectivity, how selectivity was achieved. But within this lecture, we are simply focusing on how these pesticides can be broken down. And we end there. Now, in phase 1, you can have a pesticide molecule being altered so that we have some radicals or functional groups, as they call them, being formed, like the OH, CO, OOH, SH, NH2, and CN. Now, when we have these being formed, the pesticide will start to lose its toxicity. Then in phase 2, we have got 3 specific molecules which will react with the pesticides. And we basically call these reactions conjugation reactions. Number 1, we have got glutathione, which can react with the pesticide. We also have glucose, which can react with the pesticide. Then we have got the amino acid, which can also react with the pesticide. Whatever is produced at this stage will further detoxify the pesticide. And finally, in the plants, there is a further conjugation which will even make the pesticide less toxic. So although we are applying the pesticides in the environment, we should be aware that these pesticides may not last forever within the environment. There are processes which are there. It could be the pests themselves which will start the process of breaking down these pesticides. Of course, some of these pests may die, and the byproducts are released in the environment, and they will be met with the microbes which will further deal with the processes. Even some plants may end up absorbing pesticides and further degrade these pesticides. In fact, in contaminated areas, sometimes we do plant, we can put some plants to clean up the environment. Like what happens in mines, where there is mining going on, where they use a lot of toxic products, maybe during the processes in extracting minerals. Some of the lands we have to plant either trees so that we can recover the environment which could be damaged. So the question is, what will happen to the pesticide inside the living system? Or simply, what is the fate of pesticide inside the living system? When the pesticide enters the living system, it will participate in metabolism or co-metabolism. Although it may not produce desirable results, at least there are biochemical reactions which can take place. Probably we may call this co-metabolism, where transformation makes the pesticides less toxic to the target and non-target organisms. Some of these processes which can change the activity of the pesticides are known as, number 1, oxidation, number 2, reduction. We also have hydrolysis and conjugation. Later on, we will explore what exactly happens during oxidation. Actually, oxidation could be 7 chemical reactions. Reduction, I think there are 2. Hydrolysis, there could be 3 reactions. Conjugation, probably 2 or 3 reactions. So biotransformation of an organic compound that is not used as energy source or constructive element of the organism. So these changes are not going to benefit the organism in terms of energy supply or for growth. Now, the organisms are armed with special substances which we call enzymes. These enzymes facilitate the breakdown of pesticides. I'm sure at one stage you've done digestion where a lot of enzymes are involved in breaking food so that we can get energy. So the first group of interesting enzymes which are involved in breaking pesticides are known as hydrolytic enzymes. It means they break down these pesticides. If you have esters, you can have what are known as esterases. But these are hydrolytic enzymes which are focused on pesticides carrying ester groups. If a pesticide is carrying an amide, we classify these hydrolytic enzymes as amidases. If the pesticide has got a nitrile group on it, we can call these nitrases. So if you can recognize some of these functional groups, you are likely to predict what sort of enzymes which are likely to break the pesticides. We also have two oxidases. The most common one, which is known as cytochrome P450, is the one which does a lot of work, especially in phase 1. Then we have the peroxidases, which also help to break down these pesticides, although we will focus mostly on cytochrome P450. Then reductases, nitroreductases, as well as reductive nitroreductases. And the last group is the transferases. Probably we find these in phase 2. Glutathione S transferase and the glucosyl transferase. So basically there are 4 groups of enzymes: hydrolytic enzymes, oxidases, reductases, and the last group, the transferases. So these are the products which facilitate reactions which break down the pesticides. So where do we find these enzymes which break down pesticides within organisms? Now, the cells of organisms actually contain enzymes which are capable of oxidizing pesticides. So the enzymes reside in the cells. We have microsomal enzymes which are responsible for pesticide oxidation. These are the cytochrome P450 and probably the peroxidases. Sometimes we may call these microsomal oxidases or mixed function oxidases. This is specifically pointing to cytochrome P450. Then we have enzymes in the cytosol. These are parts of the cells where the enzymes are residing, which can oxidize certain alcohols and aldehydes. Reduction processes are also done by some microsomal enzymes. Nitro groups could be reduced to amino groups, but these details we are going to see them as we proceed. Now, inside the cells of organisms, we highlighted conjugation, which can be facilitated by the membrane-bound enzyme. We have glucuronic acid conjugates, which are common in higher animals. Conjugating substances are water soluble, so some of these can dissolve in water. So let us move on to phase 1 processes. When we discussed this under weed science or herbicide physiology, we could have not mentioned some of these, but we are now looking at the whole spectrum of pesticides. So let us look at hydrolysis. What do they do? Many pesticides contain the following parts: ester, amide, or phosphate linkages. If any pesticide is either an ester, an amide, or a phosphate linkage, it can be broken down by hydrolysis. We have examples of insecticides. Most organophosphorus insecticides can be broken down by hydrolysis, which can be in insects. Pyrethroids can also be broken down by hydrolysis, which are present in insects. Carbamates can also be broken down by hydrolysis. In fact, if the insect pest has got sufficient hydrolysis, the pesticide may fail to kill that insect. And sometimes we talk of insect pest resistance to pesticides. Still, phase 1 processes. Let us focus on fungicides. Which groups can be destroyed by hydrolysis? If a fungicide is in the group dithiocarbamate or dinitrophenol, then it's likely to be affected by hydrolysis. For the herbicides, it's usually the urea and the carbamates which are destroyed by hydrolysis. So let us look more closely to the activity of the hydrolysis which are present in living organisms. The interesting part which you should look for in a chemical structure for organophosphorus. You look for R represents some carbon atoms, which are many of them, but what you should recognize is the OP. Is oxygen present? Is phosphorus present in the linkages? If it is present in any molecule, you should suspect that it's likely to be attacked by hydrolysis. But we can give special names to the hydrolysis. If a hydrolase attacks an ROP, where you have oxygen and phosphorus, we call it the phosphatases. These are phosphatases. This is further grouping. If you have got an amide, which is joined to the carboxyl or to the CO, you call this carboxyl amidases. So remember, phosphatases and carboxyl amidases are simply hydrolyses. So they are just hydrolyses, but with special names now, so that if we say phosphatases, we know they are attacking the OP. If they are carboxyl amidases, we know there should be nitrogen somewhere, NH somewhere, and CO here. Now let us look at malathion. This is the simplified structure of this insecticide. Now it can split into two, into an acid and an alcohol. And which enzyme actually facilitates this is known as the carboxylase. So the carboxylase can cause malathion to be split into two. Probably why malathion doesn't affect humans is that probably we possess a lot of carboxylase, which can destroy this malathion. I'm not saying it's a safe pesticide. Malathion is efficiently converted to acid and alcohol components in higher animals like us. This accounts for its low toxicity in higher animals like humans. We have got the hydrolysis of esters of 2,4-D in plants. We are talking about hydrolysis. The esters readily penetrate into the weed and is readily hydrolyzed to produce active dichlorophenoxyacetic acid 2,4-D. So hydrolysis can actually work on this herbicide. Many pesticides can contain these structures. The methyl group, or attached to oxygen CH3, or the ethyl group C2H5 also attached to the oxygen. I think these can also be hydrolyzed by hydrolytic enzymes. Amides. An amide is attacked by amidases, but these are also hydrolytic enzymes. What you see here is the structure of dimethoate, a common insecticide. Now, if it enters an insect, amidases are going to break it down into dimethoate acid and this CH3NH2. And this can be less toxic. So the amidases will hit here where there's nitrogen and split it to form an acid part and a CH3NH2 part. That's how it is broken down. We also have a carboxyl esterases which can attack amides. If we look at the degradation of propanil in rice, this is propanil. Now what is going to happen to propanil is that it's going to be split here by aryl carboxylamidase. Then it forms an acid and this bit. We also have a special hydrolase which is known as the epoxide hydrolase. When we talk about cytochrome P450, it also does the same thing, like epoxide hydrolase or hydrotase or just hydrase. Now, if we look at carbaryl, which is also an insecticide, it has got this structure. It has got two benzene rings which are fused together. What will happen is that at the corner here, it will be opened. Then an oxygen is added with these hydrogen atoms. And later on, it may form this hydroxyl, which is known as the trans dihydrodiol. But epoxide is just forming this part, which is interesting because it alters the other benzene ring and makes the pesticide less toxic. Now, just to reinforce your understanding of hydrolases, if we have got this structure, which is an ester, you have carboxyl esterase. If you've got ammonium bit, either CH3 or C2H5, it is the ammonia which it is the nitrogen, the NH, which tells us that it should be an amidase. Here again, it's an amidase. And these are the products. This one will produce an acid and an alcohol. These two will produce acids and amines. Now these are some of the hydrolysis. These are some of the enzymes which participate in phase 1. But we have got the role of cytochrome P450, which by far does a lot of changes to the pesticide. This one is really involved with the pesticide degradation. We have got 3 major processes which can be facilitated by this enzyme. We have got oxidation, hydrolysis, and reduction. Oxidation could be 7 reactions which we'll look at. Hydrolysis, maybe 3 reactions. Reduction, maybe 1. Then conjugation, maybe 3. But when we talk of conjugation, we are now in phase 2 and 3. So like I said in my introduction, I said that if you take a pesticide, put it inside a pest, some of these reactions are going to take place. Phase 1 reactions will have oxidation, reduction, and hydrolysis. Phase 2 reactions, we are going to have conjugation. Even in phase 3 reactions, this is just conjugation. But there's going to be mobilization of the conjugates into vacuoles, and some of the conjugates are going to be part of the cell walls. This is now happening in plants. So let us focus our minds on the cytochrome P450 monooxygenase, which occurs in the endoplasmic reticulum. Sometimes we call this the monooxygenase system. Other names are mixed function oxidases or microsomal oxidases. They speed up oxidation reactions by simply inserting oxygen into the pesticide molecule. That is their function. They take oxygen, put it in the pesticide molecule so that the pesticide molecule gets oxidized or destroyed. The oxygen atom may be converted into water by the electron donor NADPH. So basically this is the activity of cytochrome catalyzed reaction. We will just give you one reaction so that you understand what it does to all the pesticides. It requires NADPH. It also requires hydrogen. It requires oxygen. And this should be the pesticide. If you have these reactants, you will get NAD, which means NADPH will lose its hydrogen. Then it forms water, and this pesticide will gain an OH, which means that it will cease to be the pesticide. So RH represents the pesticide, ROH represents the pesticide metabolite or the changed pesticide. We have got what are known as xenobiotics. This can be just foreign substances which can enter the organism, which we call a pest, and it can be acted upon by cytochrome P450. Now, cytochrome P450 helps in the formation of these functional groups which are added to the molecules. If I go back a bit, if you take this RH, you can see the OH was added. The OH here is what we call the functional group, and there are quite a lot of functional groups depending on the molecular structure of the pesticide. So these are some of the examples: NH2, SH, COOH, CN, OC, H3, CH3, SC, H3. And we said these are functional groups which are added to pesticide molecules. As a result, there is a reduction in phytotoxicity. Okay. Someone wants to end. Oxidation reactions. Now, I said cytochrome P450 can support oxidation reactions, and we have 7 reactions. The first one is known as hydroxylation. This is not new to us. We have got N-dealkylation, O-dealkylation, oxidative deamination, sulfoxidation, or thioether oxidation. 1 to 5 we discussed them under herbicide selectivity. Now we are going to add epoxidation and the replacement of sulfur atom by oxygen. And please remember that these reactions are facilitated by cytochrome P450. Here is the first reaction, which is hydroxylation. If a molecule carries a halogen or a chlorine atom on it, it can be in this format if it is an aromatic group, or if it is an aliphatic group, it can carry this chlorine. So chlorine is the key to hydroxylation under the cytochrome P450 activity. Now this can be replaced by a functional group which is known as the OH. Either it could be on the benzene or aliphatic group. So please remember that if you observe a chlorine atom on the structure, on the chemical structure of any pesticide, suspect that hydroxylation can take place as a way of destroying that pesticide within a living system. So here we have got pyrethrin, which is an insecticide. What can happen is that on this pyrethrin you can have an insertion of the OH. Then you have what is known as hydroxypyrethrin, which means it loses its toxicity. Here we've got Basagran. We can have an insertion of OH group on this part, or we can simplify it like this, where we show that OH is now part of the new molecule, and therefore it loses its activity as a herbicide. On the other hand, if you take an insecticide, carbaryl, now with the carbaryl you can insert OH in any position of the benzene ring. In fact, we have a mixture of products depending on where OH has been inserted on carbaryl. This could be happening in an insect because the carbaryl is an insecticide applied to insects, and when the carbaryl enters the insects, these things can start happening. Once carbaryl has got an OH, then it's no longer useful. The other process is known as N-dealkylation. If there's an NH, this R represents the complicated part of the molecule. So we focus on what is important, NHCH3. If you have NHCH3 in any molecule which is complicated, suspect that this can lead to this chemical reaction, N-dealkylation, or the removal of the ethyl group, leaving an amine. Well, we can look at atrazine, which has got such structures. This part, because it has nitrogen, it can be changed to NH2, or we can simplify it here, where we have N attached to this structure. This is going to be split, then we have a RNH2, and atrazine will cease to be the herbicide inside the plants. Diuron. Now, diuron has got this grouping. This CH3, this can be removed. Leaving the H here. And you have this, the formation of the OH is now on the methyl part. Once you remove CH3, this will cease to be the herbicide. The other process apart from any dealkylation is where you have oxygen instead of nitrogen. But the description of the process is simply the same, except that we now have oxygen instead of nitrogen. attached to this arrow, which I said is representing a lot of complex structures. So this CH3 can be removed, leaving the OH, and we call this O dealkylation. This is 2,4-D, that's what it looks like. O dealkylation can take place where the arrow is pointing, and you will end up having an OH here. Here we've got methoxychlor. I think this is an insecticide. Here we can affect this part because it has oxygen and CH3. You know, like I said, if you have this is the key which will tell you what sort of reaction is going to take place. It's O dealkylation. So it's here, it's also here, and it will form this product. If you have an S sulfur attached to CH3, or wherever you have sulfur in the system, suspect what is known as sulfoxidation or thioether oxidation, because the result is that cytochrome P450 will support the attachment of oxygen to the sulfur, and the pesticide loses its activity. We have a prometrine, we have got sulfur somewhere here, and it will gain, on the other hand, it will gain oxygen, and you call this sulfoxidation or thioether oxidation. Here we've got EPTC. It's the herbicide which is used to control Cyperus esculentus in maize. Here where we've put a green circle, we are showing the position of sulfur. And after sulfoxidation or thioether oxidation, we now have a second oxygen being inserted into the sulfur. Then this herbicide will cease to be active, and we have simplified the reaction here. We are simply showing that oxygen has been added to the position of sulfur. There is what we call oxidative deamination. In this case, you have your R, which represents something which is complicated, but you have got two nitrogen in sequence, NH attached to NH2. If you have such a situation, after oxidative deamination, we are going to remain with the one NH. And the example could be this Metabolism. It is this nitrogen and the other nitrogen here. So this will go away during oxidative deamination and leaves two hydrogen atoms attached here. I mean, if you look here, we have got an S. There is also a potential of sulfoxidation here, or thioether oxidation, by simply adding oxygen there. So these things are not that complicated. It's just the art of seeing what this is representing, what this is representing. Reduction, if you are going to have a reduction, you should have an R with NO2. This is probably nitrate. This can be reduced to NH2. Like what can happen to trifluorine, which is the herbicide which is applied to the soil, then incorporated. It carries two nitrate components. This can be changed to an amine. The same thing here can also be changed to an amine. And we call this a reduction process. Now, to be specific, we say nitro reduction, meaning that we have got this nitrogen here, or the nitrate. This is now a new process. Reduction is one reaction. Epoxidation, you have this double bond here, and it is going to open up, and we put oxygen. Once you have this process, we produce an epoxide. I think it could happen to Cabri. Anyway, we've got this aldrin with this double bond here. This can open up, and we put oxygen there and form dieldrin. So from aldrin to dieldrin. Dieldrin has got oxygen where we have opened this bit, and we call this process epoxidation. And when you are talking about it, there's no need to remember this. In fact, it's not possible for you to remember these structures. But probably you can remember this, or you can remember oxidation, epoxidation, meaning you are simply adding oxygen. Now there is also what we call a replacement of sulfur atom by oxygen. You can have a structure where you have a phosphorus and sulfur. This sulfur will go away, and it's going to be replaced by oxygen. So it's simply a replacement of sulfur by atom by the oxygen. It can happen to parathion, where you have sulfur here. Maybe this is less toxic, but paraxon could be highly toxic. You may have to check. If we remove sulfur and put oxygen, we make this more toxic, I think. And if we apply this to insects, those which can convert parathion to paraxon, they are going to die. They will not survive. You may have to check my statement, but that's what I think should be happening. Of course, sometimes we make the pesticide less toxic, but sometimes we elevate its toxicity. And this could be an example, but you can check and give me the feedback if it is otherwise. Now we are moving on we are now through with the oxidation, which has got 7 reactions which we have gone through, starting with the hydroxylation, ending up with the substitution of sulfur with oxygen. Then the reaction of the pesticide with water is what we call hydrolysis, and it can cause the breakdown of the pesticide into an ester, a nitrile, and an amide. Here is a situation where we have an ester. An ester is a carbon followed by 2 oxygen atoms attached to the others, which represent other things. This can be split into an acid and an alcohol. It's called ester hydrolysis. We can have a 2,4-D methyl ester, which can break down into an acid and an alcohol. And we call this de-esterification. In some plants, it can produce this acid, which can also elevate the toxicity of the herbicide. Here we've got amide hydrolysis. If you have an amide where you have NH, then carbon, which is attached to the oxygen. So the key is to recognize NH attached to carbon, then a double bond with oxygen. This will lead you to amide hydrolysis. So if we give you a chemical structure, you should be able to recognize this structure so that it tells you how that molecule is going to break down, at least via amide hydrolysis, and it will form RNH2. Propanil has got also the amide. You can see I have circled it. This is only what you should remember, not the rest of the molecule. And it can break down into an acid and an amine. Hydrolysis of the triple bond, where we have carbon to nitrogen. This is the cyano group. So you need to recognize this triple bond so that it leads you to the hydrolysis of a cyano group. So here the cyano group, first it will form an amide, then the end products are acid and ammonia. So if you get an acid and the ammonia, that's nitrile hydrolysis. We can get it in Bladex or cyanazine. We just focus where we have N and the carbon here, and the triple bond cyano group. It can form an amide, but later on the end product could be an acid. The rest of the molecules are not important. Again, bromoxynil, bactrial, we just focus on the triple bond or the cyano group, which can liberate a carboxylic acid and possibly an amine after hydrolysis. So this is the hydrolysis of the cyano group. Now we've finished with the phase 1, which is a bit long. Do you have any questions at this stage? So we move on to phase 2 reactions. We have got conjugation reactions. Here we are interested in glutathione. This is the chief actor in this phase. It's going to utilize products from phase 1, and it is going to be facilitated by glutathione S transferases. Or we can just call it GST. Even if you just in an exam you call it GST, we know what you are talking about. Now on glutathione we have got the SH, which is the reacting component. Glutathione GSH, it is a tripeptide of 3 amino acids, namely glutamate, cysteine, and glycine, or we can shorten them like this: glu, cis, gly. Here's an example of a simplified conjugation reaction where you have got the herbicide now carrying the OH reacting with the SH component of glutathione, forming this conjugate, glutathione conjugate. Once you have this taking place, it means the pesticide has been fixed. It's no longer going to be useful. Now, glutathione conjugates could be transferred to the vacuole for storage if it is in the plants. If we have an epoxide which had been formed in phase 1, glutathione likes epoxides, these structures. So it is going to get joined to that and fixes the pesticide. Again, it likes the chlorine atom, so it can get joined to one of the chlorine atoms and form this bond. So the presence of the epoxide, the presence of chlorine in any molecule will attract the glutathione to act on it. Also, the presence of this phosphorus attached to the oxygen in diagonal, for example, it can be split like a scissor. So this can be split by glutathione, and it will form a union with the other part. So you need this structure so that glutathione can act on it. The other structure is an iodide, like here where we have iodine attached to this, and you can have SG, notice Secretary General. So the SG can be attached to the CH3. Or you can have, although there's a potential to, in methyl parathione, there's a potential to cut this molecule here, but we can also split it from this end and form methyl attached to the SG. And this may involve glutathione as alkyl transferase. we know from our previous experience that glutathione can directly attack atrazine at the chlorine atom and form conjugates, which later on reduce the toxicity of atrazine in maize, and this is facilitated by glutathione S transferase. DDT, because it has these, chlorine atoms, one of the chlorine can be attached to SG, and this can detoxify the DDT, which is quite an old pesticide. Again here, because we've got a chlorine, we can have glutathione being attached to where chlorine is and detoxify the pesticide. Here is a summary of conjugating agents. We have got glucuronic acid in mammals, birds, reptiles, terrestrial amphibians, and some fish. Glucose, higher plants, insects, and mollusks. Arginine for arachnids and myriapods. Glutamic acid or glutamine, again arachnids and some myriapods. Then glycine, fish, crustaceans, and we can also have sulfur as a conjugating agent, again in some amphibians, some crustaceans, and so forth. Just briefly on the role of glucose and glucuronic acid. Glucose and glucuronic acid combine with some of the pesticide products formed in phase 1. And this is further reducing their toxicity. The donor of glucuronic acid is uridine diphosphoglucuronic acid, UDPGA. But for glucose, the donor is UDPG. Let us look at the difference of glucose and glucuronic acid. It's here. Here you've got an alcohol in glucose. In glucuronic acid, you've got an acid here. That's why we call it glucuronic acid. But they share more or less the same chemical structure. Sugar conjugates. Now, sugar, we know it is required for energy within a living system, but it can form, it can detoxify pesticides. So sugar can combine with pesticide metabolites if they have an amine, an adenamine or carboxylic acid or a phenol. If any pesticide has these, it can react with the sugar to form sugar conjugates. So uridine diphosphoglucose, UDPG, is the sugar donor. UDP glucosyl transferase is the enzyme which facilitates this process. So UDP glucose is the sugar donor. Here is a simple reaction which is known as O-glucosylation. You have the herbicide carrying the OH. The UDP glucose, which is the donor, will release sugar so that it combines with this part of the herbicide and it leaves the process, and the whole process is facilitated by glucosyltransferase, and it is called the O-glucosylation process. Here is a real example of sugar donated to Basagran, the herbicide which you know, and it forms Basagran joined to glucose, and Basagran becomes detoxified. If the herbicide carries an NH2, again UDP glucose will donate the glucose to this part of the herbicide. Then you have glucose N herbicide being formed, and you need glucosyltransferase to facilitate the process. We have got the parathion, which can also be detoxified by adding glucose to this pesticide, and UDP is released in the process. If you have got a COOH, you have glucose ester formation. Again, glucose is added into the process, resulting in the formation of an ester. Glucosyltransferase is required as well. This can take place in 2,4-D, right? Where glucose is actually added and it forms glucose ester of 2,4-D. Here is a case of glucuronic acid, which can be added to carbaryl at two different positions. It can be added here, or it can be added at this position. Amino acid conjugations. You can have a herbicide with a COOH and aspartate, and they can give us herbicide which is combined to the aspartate, and the herbicide gets detoxified. Here's a real example, 2,4-D here, and aspartate on the other end. Then you have this complicated structure being formed. Now we have got sulfate conjugation. You need the sulfotransferases, and you need the 3-phosphoadenyl 5-phosphate or PAPs. If you have these structures in any of the pesticides, you end up having this RS with these 3 oxygen atoms attached to it. This is sulfate conjugation. This is less toxic and more soluble in water. before I go further, it's these structures which are affected by sulfur. Phase 3 reactions. Plants do not excrete pesticide metabolites. There can be further conjugation of phase 2 products, moving metabolites into vacuoles and storage, incorporation of metabolites into cell wall region, pesticide glucosides further processed by the addition of malonic acid residue to sugar conjugates. Okay, so you can have this could have been formed in phase 2. Let us get this one right, where glucose get joined to the herbicide. But malonate can also be added in the presence of malonyl coenzyme A, so that you form malonyl glucose herbicide, and coenzyme A is released. Now this part here is incorporated into the cell wall, or it can go into the vacuole. For example, you can have a metribuzin attached to glucose, and the glucose can be added. You get metribuzin, glucose, agent biocide. So this is it. Let us have the summary of pesticide metabolism. If you are interested to know what we have just gone through, this is the summary. You need to know the hydrolysis, what they do. I will not go through them again. In phase 1 processes, please note that we have got oxidation, reduction, and hydrolysis, and the chief actor is P450, cytochrome P450 oxidative enzymes, and there is formation of these functional groups, like what we have said. And again, this is a summary of what is happening, and all the reactions are also described here. We have got reduction, which is one process. Hydrolysis, there are 3 reactions which are also summarized here. This is just a summary for you, so that you know where you are going and where you are coming from. Then conjugation processes, where you have the role of glutathione, the role of amino acids, and the sugar. I will not repeat what I have said. Again, phase 3 reactions is actually addition of malonate to herbicide sugar conjugates. And again, a summary of what is actually taking place. This is the summary. At the end, we have got the summary of the whole process, so that you can spot in key issues. Actually, that is where the questions may come from, at the back end there. So this is it. Let me close this. I will stop streaming. Are we all here? Hello?
Yes,
Hello, Doc.
yes, we are here.
Yeah,
I,
yeah, tiripo,
I thought
Doc.
I was alone.
Tiripo.
Yeah, so this is
Yeah,
it
we're there, Doc.
on pesticide metabolism. If you see a question on it and you think you can do it, because you can get all the marks, right? Normally I split the questions into small units. Do you know hydroxylation? If yes, what does it look like? Can you just show us what hydroxylation is? Or dealuculation and dealuculation? Very simple things, not remembering the chemical structures. And we may give you some molecules so that you can identify the structures where things are happening, right? And tell us what is likely to happen to the molecule. And you get your marks. Any questions? You've done some of it, isn't it? When you were doing with the science. But in with the science, we're emphasizing on herbicide selectivity. Here we're just interested in breaking down of the pesticides, either in insects or in the plants. And I've tried to simplify it can be a very complicated, biochemistry subject, but we tried to minimize certain issues. So we'll meet on Tuesday night, is it? Both Monday and Tuesday, during the day we have a workshop. But on Monday I can't give you a lecture because I'll be in the middle of a workshop. After the workshop on Tuesday, then we continue with the other examples of pesticides. So far we have addressed one question, right? Which is coming actually. But there are plenty of questions. You have a choice. Any issues? So I think after this section I will give you an assignment. I will think of an assignment, then I will post it so that it will help you to revise this very important section. Any questions? I think I've talked too much, isn't it? So I am now switching off here. Muchirimu area kumarara.
Thank you, Doc.
Saka tosangana ne Tuesday night handiti.
Okay.
Tombodhonza toona kuti tosvika papi, but I think we may need our target is to have 2 weeks. Then we have 1 week yekurevise handiti. Then the 4th week maakunyora. So we target the lectures for the first 2 weeks. The 3rd week we target it to revision. 4th week you write and finish. that is the plan. Kwete kuti taakupinda mumaexam kuti toda kuti tipedze zvinhu. You need one free week which you can focus on your revision. Tikangoita those 2 weeks ma-assignments edu tichiita and so forth, you'll find that kumberi uku zvonokuitirai zvakanaka, because hatidi kuti muomerwe muri mu-ACH muno. Just go there, do what you have to do, then you proceed. Okay, so let us communicate. I am now, I need to stop the recording as well.
Thank you, Doc.
Mune lecture ningi video, but irori ndogona kutumirai futi.